I will start my presentation now. Okay. Can you see this one? This is the pictures showing the development of dicot embryo. But before we start this development of embryo and all, let me revise a little. Okay. What we studied last time? We studied double fertilization, then types of endosperms. First, we'll recap a little about the double fertilization. What is double fertilization? That the fusion of one of the male gametes with the egg cell, that is the female cell, female gamete, that is called as syngamy or first fertilization, that results into zygote. And the second male gamete fuses with the secondary nucleus, that is the definitive nucleus, which is already deployed in nature, that results into the formation of TPEN, that means triploid primary endosperm nucleus. So second, this is called second fertilization. So at two places, twice this fertilization takes place in angiosperms and both the male gametes are utilized. So this is called as double fertilization. Okay. So the as we discussed last time, that the result of double fertilization is what? The result of double fertilization is, first is the zygote and the second is the endosperm. Okay. So we are, last time we have studied the different types of endosperms. Okay. Now we will come to the next, that is the result of fertilization itself, as the embryo. So as everyone knows that first fertilization, that is syngamy, results into zygote. Zygote is the result of first fertilization, and the second fertilization, that is the formation of endosperm. And last time we have discussed the development of endosperm and its types. Now today we will discuss about the development of embryo. That is what? The result of first fertilization. So what is the result of first fertilization? Zygote. Zygote, that is, the process is syngamy and the result of first fertilization is zygote, and that in the further development gives rise to embryo. Now let us see what is the definition of development of embryo. Means what is the embryogenesis? The process of development of zygote into an embryo is called embryogenesis. The embryo is developed at the micropylar end of embryo sac. And as you studied last time, that endosperm, that is produced at the chaliceal end. So remember these points. Why I am telling? Because the further explanation is related to these 2 points. That means zygote, which is the result of first fertilization, gets converted into embryo, and that embryo development is called as embryogenesis. So the process of development of zygote into embryo is called as embryogenesis. Genesis is synthesis, that is formation. Okay, genesis is synthesis or formation. So how this embryo is formed? That is from zygote. And what is the zygote? That is the result of first fertilization. That means fusion of one male gamete and the egg cell. Where it is present? In the embryo sac towards the micropylar end. Whereas the result of the second fertilization, that is the formation of endosperm, is towards the chaliceal end. Remember these points because our next explanation is related to these 2 ends, micropylar end and chaliceal end. Now what happens with the zygote? Zygote first of all forms a wall around itself, that is called as oospore. Then in its further development, it shows the proembryo, formation of proembryo and then embryo. The previous stage of embryo is called as pro-embryo. Now, how it takes place, that we will see. First of all, I will explain this with the help of these points, and then we'll come to the diagram. Okay. So, as I said, zygote forms a wall around itself and it produces oospore. Now, this oospore, by mitotic division, that means mitosis, divides in two unequal cells. Try to understand. Now we are talking about the zygote. Okay, zygote, by forming a wall around itself, forms oospore, and that oospore divides mitotically to form two unequal cells. Now, which is larger and which is smaller, that also you need to remember, because the further development is related to the development of embryo with these two important cells. Okay, so larger cell, this is also called as basal cell, is called suspensor cell. Larger cell which is towards the base, that means which is towards the micropylar end. The cell which is towards micropylar end is large in size, and this is called as suspensor cell, or this is initially called as suspensor initial. And the smaller cell which is towards the chaliceal end, that is terminal cell, called as embryonal cell. Try to understand, because I will be using these two words only. Larger as suspensor initial, that is going to result into suspensor cell, or later stage suspensor, and the smaller cell which is the towards chaliceal end, that is embryonal initial, resulting into formation of embryo. So what is suspensor and what is embryo? That embryonal cell, that I will show you in the diagram. But right now you remember that zygote by the mitotic division results into two unequal cells. Larger cell is towards micropylar end, called as suspensor initial. Smaller cell is towards the chaliceal end, called as embryonal cell. Now, suspensor cell, now I'm going to tell you, explain you both the divisions for the further development of both of these cells. But at a time, I can't explain two, so one by one I'll be telling. First, talk about this larger cell, that is the first, which is towards micropylar end. So this suspensor initial, this is first of all, it is called as initial, divides transversely, and a row of 6 to 10 cells is formed, and that is called as suspensor. Try to understand. So the first larger cell towards micropylar end, called as suspensor initial, divides transversely to form a row of 6 to 10 cells, and that is called as suspensor. So last cell of the suspensor becomes large and swollen. It becomes larger than the remaining cells and it becomes swollen. That is called as hostorium. Whereas the first cell of suspensor, in the suspensor, that is called, which is nearest to the embryonal cell, that is called as hypophysis. And what is the role of suspensor? The role of suspensor is to push that embryo towards the endosperm for the nourishment. So, now simultaneously, when the suspensor initial shows its divisions, simultaneously this embryonal cell, that means the smaller cell, that which is towards the chaliceal end, that also shows its divisions. It also divides mitotically. 3 consecutive successive mitotic divisions takes place. So first division, that is the smaller cell, embryonal cell divides. So with the result of first mitotic division is formation from that cell, 2 cells are produced. Second mitotic division, 4 cells are produced. That is called as the quadrant stage. Then the third mitotic division, 8 cells from 4 cells, that is the quadrant stage, gets converted into octant stage, that is called as octant embryo. So this octant embryo is also, up to this can be considered as the pro-embryonal stage of embryo. Means before the formation of embryo, whatever the stage, this is the initial stages of development of embryo, that is considered as pro-embryo. Now, what happens with this? Now, little bit different type of divisions take place in embryonal cell. In suspensor cell, straightway transverse division takes place and a row of a 6 to 10 cells is formed that results into suspensor. But in case of embryonal initial, what happens? The first cell, first mitotic division, then second mitotic division, then the third one. All these mitotic divisions, they cells are taking place in right angle to the previous one. So in that particular plane, planar division, right angle to the first division, they are taking place. So the 8 cells, that is the octant stage of embryo, is developed, formed, and lower tier of 4 cells of octant gives rise to hypocotyle, which in the further development gives rise to radical. And the upper tier of 4 cells of octant gives rise to epicotyle, which in further development gets converted into plumule and 1 or 2 cotyledons. Now listen. Here, up to octant stage of embryo, that is the pro-embryonal development, in case of dicot as well as monocot, is similar. In the later stages, little developmental changes take place in monocot and dicot. In case of monocot, only 1 cotyledon is developed. In case of dicot, 2 cotyledons are developed. Okay, so this you remember now. Up to octant embryo, that is the pro-embryo development, up to pro-embryo development, it is same in mono as well as dicot. Now, octant embryo in the further development gives rise to globular embryo. The shape is globular, so that's why it is called as globular embryo. Then it in the further development gets converted into heart-shaped structure. And then in the further development, it gives rise to horseshoe-shaped structure. So that's why these are the names given as per the shape of the embryo, nothing else. Because the suspensor development is clear. Suspensor initial straightway transversely divides, forms a suspensor. But here in the upper cell, that is embryonal initial, the developmental stages are little bit different and main. That's why all these different forms you can observe. Octant embryo gets converted into globular first. It looks globular in shape. So in the further development, it looks like heart shape. And in the further development, it looks like horseshoe shaped structure. So at the end of the development of embryo and endosperm, the ovule gets converted into seed and ovary into fruit. Now I will explain with the help of diagrams, which first of all I showed you. Okay, so I'll come to the textbook again so that I can show you the textbook diagrams. Now you come to this. Okay, little bit. See, first diagram now. This is labeled as A. What is this? This is the, you can see the near this arrow, this is called as the zygote, which forms the wall around itself, so called as oospore. Now in the second B, that is the diagram labeled as B, you can see this zygote, that means oospore, divides unequally. So you can see this is smaller cell and this is the larger cell. So smaller cell is towards the chalizoid end and the larger cell is towards the micropylar end. Okay, so this is upper chalizoid cell is called as embryonal cell. This lower cell, which is towards micropylar end, is called as suspensor initial. Embryonal initial and suspensor initial in the initial stage. Okay, now here you can see simultaneously the divisions in both the cells are taking place. In this diagram you can see here the first division of embryonal cell is also shown, and here S1 S2 is also shown, that is the division of suspensor initial. Now in this diagram, see, you can see this here, the next stage, here from 2 cell, here 4 cell structure of embryonal cell you can see, as well as this transverse divisions of all the suspensor cell you can see. So this lowermost cell of the suspensor, which becomes swollen and enlarged, this is called as hostorium. In case of suspensor, you can see this is the entire suspensor, and the lowermost cell of the suspensor, which becomes larger in size and swollen, this is called as hostorium. Okay, and the uppermost cell, which is immediately to the embryonal cell, next to that embryo development is, embryonal cell is developing. This is called as hypophysis. Okay. Now, here, one more thing you have to understand, that here you can see only 4 cells. That is the quadrant stage. But in the further development, that is the octant stage also, as there the divisions are taking place at right angles to the previous one, here, this is just 2D diagram. It's not 3D, 4D like that. So you can see only the front side, only one side. So that's why you are looking here only 4. But 4 are at the back side. So total you can consider it as the 8 cell structure as octant embryo. And up to this, the development of the embryo and the dicot as well as monocot is similar. Now, for your better understanding, you can look at this diagram, that is the next diagram labeled as D. This is the next diagram, that is D. Now, you can see here that this is the entire structure of ovule. You can see the integuments here, 2 integuments, outer integument, inner integument, and this is the funicle, and this is the, you can see the opening called as micropylar opening, and here is the entire structure of suspensor. So suspensor, you can see the lowermost cell, which is nearest to the micropyle, has become swollen, larger than the remaining suspensor cells, right? So it goes faster first in the first development, previous stages of development. So suspensor shows like this, okay? So this is the last lowermost towards the micropylar end is hostorium, and the uppermost cell of suspensor is labeled as H, that is hypophysis. And you can see this is the embryo, embryonal cell development of embryonal cell. Okay, you can see this octant stage. In the further development, it gets converted into globular structure, and then it gets converted into heart-shaped structure. Now you can understand now here the role of suspensor. When this suspensor, what is the role of suspensor? As I explained you, that it pushes this embryo towards the endosperm. Why? Because here is the development of endosperm. As last time we discussed different types also. Here is the endosperm. And what is the role of endosperm? It is the nutritive tissue. So for the further development of this embryo, nutrition, whatever nutrition is required, that is taken from endosperm. So this suspensor pushes this embryo towards the new endosperm so that it gets the nourishment from the endosperm. Endosperm is nothing but the nutritive tissue. And this nutritive tissue, that means this tissue, endosperm, is providing nutrition to the developing embryo. Now, this octant stage in the further development gets converted into globular structure, and then it gets converted into heart-shaped structure. Now you can see this heart-shaped structure here, a single development of embryo is shown. That means this lower part as suspensor and the upper part as embryo. Okay, so globular structure now has got converted into heart-shaped structure. You can see in this diagram labeled as E. This is diagram E. Only the embryo to entire embryo embryonic development is shown. So here this is the heart-shaped structure of embryo, and this is the long chain transversely arranged cells with the largest swollen cell at the end called as the hostorium. Okay, now you can see here in the next stage, next F diagram, that is here again you can see this, whatever the space was free here, that now as the embryo is developing, the size of the embryo is becoming larger. So it is enlarged than the previous one. If you see this structure, octant and embryo, and if you see this structure in the further development of embryo structure of embryo, the embryo is getting enlarged. So occupying the space of what? Endosperm. Okay. So heart-shaped structure in the further development gives rise to horseshoe-shaped structure. So it is shown this horseshoe-shaped structure along with the further development. So in sometimes in case of endospermic seeds, the entire this some part, most of the part, half of the part, more than half of the part is utilized by this embryo, and then in the other type that we are going to study again in the next part, some part is remaining, and in some cases the entire endosperm is occupied or utilized by this developing embryo. Now this is this diagram of the entirely developed embryo. Now you can understand now the role of suspensor has got over, because it has already pushed the embryo towards the endosperm for its nourishment and the proper nourishment. Because of the proper nourishment, that embryo has enlarged properly, developed properly, and so this has become now how it looks very small. So you can see in this diagram the further development in the horseshoe shaped structure. In case of dicot, here you can see two cotyledons, hypocotyledon, which gives rise to radicle, and radicle gives rise to root in the further development. Root structure, that is the radicle in the further development, gives rise to root initial, that is covered by the cap, that is called as root cap. S is nothing but the suspensor. It afterwards becomes very small and then disorganizes. And here this structure which is above the cotyledon, this part is called as epicotyle, which gives rise to, you can see here, this gives rise to plumule, which in the further development gives rise to shoot apex. So hypocotyle gives rise to radicle, that gives results into the formation of root, which is covered by a structure is covered by a cap like structure, that is the root cap. Whereas epicotyle, which is above, epicotyle is above the cotyledons, this structure will give rise to plumule, that results into the formation of shoot apex. I hope you have understood up to this. Now, what is the difference between the dicot and monocot? Little difference is there, that I will show you in the next diagram with the help of this. See, this diagram I am not going to explain you that development of embryo in monocot, because here the cells which are shown, they are oppositely shown. That means in this diagram, why I am not explaining this diagram purposely, I am not explaining because the first up to octant stage, this is common, development is common, and here opposite these things are shown. That means embryonal cell should be towards the micropylar end. Here the micropyle embryonal cell is towards chaliceal end and suspensor cell is towards the micropylar end. But here it is considered as orthotropous ovule. But what we are studying is anatropous ovule. So in angiosperms, anatropous ovule is a characteristic feature. So that's why when you consider all these diagrams, all these up to E or F diagrams, in the opposite direction, means suspensor you show lower side and the embryonal cell you show on the upper side, then only it is correct. So that's why I will not explain you this part. Already in the previous, all those diagrams of development of dicot, it is very clear. So I will not come to this diagram, these all diagrams, but now directly we'll come to this diagram, that is the mate seed, how it results, how that is formation of this mate seed. Now what is this? You just try to understand. Okay, see, in case of maize is the example of monocot. Okay, monocot. So only single cotyledon is observed. Now where is that cotyledon? In monocot embryo, single cotyledon occupies the terminal position and plumule is lateral. And this single shield-shaped structure, that is cotyledon, is called as scutellum. This is the cotyledon, and this entire structure is called as endosperm. Here it is very clearly labeled. I will show you the video also at the end afterwards. So first try to understand with this diagram. So in case of monocot, single cotyledon is observed and this shield-shaped structure is called as scutellum in case of monocot in this maize. And this is the endosperm. So some part of endosperm is also there and only single cotyledon is developed. And in this structure you can see this is the plumule and this is the radicle. Plumule is covered by coleoptile and radial is covered by coleorhiza. That means the shoot cap and root cap, they are also called as shoot cap, means the initial part in the initial stages, it is covered by the structure called as coleorhiza in case of root. Whereas in case of shoot cap, that is the plumule, which in the further development gets converted into shoot structure. First of all, in the initial stages, it is covered by coleoptile. So coleorhiza covers radicle, coleoptile covers plumule. So this is the simple structure of maize seed. Where one more thing you have to remember, that this sheet step structure is the single cotyledon, and you can see even this part of the endosperm. This is the seed coat fruit wall, that is, and this is the one layer you can see that is called as aileron layer. You can see this diagram. Okay, so that is only the difference. Now one more thing I just want to mention, that is about the uh, coverings of the seed, that is outer and inner, that I will show you with the help of remaining part. I will explain with the help of slides. Okay. This part we have already discussed, that this I will not explain, as I said. Now seed and fruit development. First we'll talk about this point, and then we'll come to the different types of seeds. Seed, the goal of reproduction in every living organisms, including plants, is to create offsprings for the next generation. Seed development is initiated by fertilization. That means what? First fertilization, syngamy, resulting into zygote, which in the further development gives rise to embryo. And the second fertilization, that is triploid, primary endosperm nucleus, in the further development gives rise to endosperm. So both together gives rise to what? Seed. So ovule, the entire structure gets converted into seed, and the entire ovary gets converted into fruit. So what about these two parts, that is the integuments? You know, I showed you in the diagram that outer integument, that is called as outer seed coat. The terminology used is called as testa, and inner integument, that is the inner seed coat, that is called as tegmen. So ovule gets converted into seed and ovary gets converted into fruit. Now you have to remember these 2 points also, that is endospermic seed and non-endospermic seeds. Okay, endospermic seeds means what? Here endosperm remains conspicuous and fills a greater part of seed, called as endospermic seed or albuminous seed. Okay, for example, castor, coconut, maize, etc. Now you remember in this thing, I showed you the maize diagram purposely, and I showed you the endosperm area part also. That means what? Endosperm was still present. It was observed. So that seed where endosperm is still, it remains. It is not totally utilized. So that is what called as endospermic seed. I showed you in the diagram of maize, right? So that is also called as albuminous type of seed, endospermic or albuminous seeds. And the second is what? Non-endospermic seed. They are also called as exalbuminous seeds, where endosperm is completely utilized by the developing embryo. So there is no endosperm remain at the end. It doesn't remain at the end. So that means what? The embryo absorbs food reserves from the endosperm completely during its developmental stages, and so endosperm disappears in mature seeds. The resultant seed is called as endospermic or albuminous type of seed. So you remember, and the examples are all pea. Now, and all the leguminous, bean, pea, etc. These are the examples, and all the leguminous plants you can remember, they are the non-endospermic seeds. Legume is what? Pod. All these pods, and most of the monocots crops, they are of endospermic. And beans, peas, etc. These are the example of non-endospermic seeds. Okay. Now, what is the significance of seed and fruit formation? Fruits provide nourishment to the developing seed. You know, fruits provide nourishment to the developing seeds. Fruits protect the seeds in immature condition. Seeds are protected. And why? Because they are very important. Why they are important? Because they are the propagating organs. So seeds serve as propagating organs of plant. That means from this, that seed is only having the ability to give rise to a new plant. So that's why these seeds are very important. So they are protected by the fruits, means its cover. And seeds and fruit develop special devices for the dispersal and help in the distribution of species. These seeds, they are carried, fruits are carried by different ways, different devices, and that's why that dispersal also plays very important role in the distribution of different types of species. That's why these, because of this dispersal, maybe by insects to some extent, maybe by birds, animals, or there are different devices used. So because of this dispersal of seeds and fruits, these species are distributed in different areas and we can get the varieties. Okay, so this is what the significance. Now, few more terminologies remaining in this chapter we are going to discuss, and at the end I will show you the videos and some questions. Dormancy. In case of seeds, this is very important point. Dormancy in general, just remember word as resting phase. Dormancy is nothing but the resting phase where the seeds are inactive. They remain in the inactive form. That means structural and physiological adaptive mechanism for the survival of the seed is called dormancy. This is the definition. But how will you remember what dormancy means? What? Dormancy is nothing but the resting period of the seed, where the seeds do not germinate. They remain as a, though favorable conditions are available, they will not germinate until and unless they take proper rest. You people take the rest and then only you are active, right? The same way, the seeds also, they take the rest and then they are ready to germinate. That, and each and every species dormant period is different. Okay, mature and viable seeds will not germinate even in presence of favorable conditions. The viable seeds germinate only after completion of dormancy period, and dormant period varies from species to species. You remember this thing. Now, few more 2-3 terminologies are remaining in from this chapter that we will discuss. Now, next is what? Apomixis. It is the phenomenon of formation of embryo or embryos through a sexual method of reproduction without formation of gametes and the act of fertilization. Embryo develops in the ovule and ovule develops to form seed. Now, there are two important points we need to remember in this case, and that is, in apomixis, mixis is what? Nothing but the mixing. What and how it takes place, that you have to remember. Okay, there are 2 points you need to remember in case of apomixis. When gametophyte organ or cell produces embryo like structure without fertilization, this is termed as apogamy. And similarly, when diploid sporophyte cell produces a diploid gametophyte vegetatively without meiosis, is called as aposperm. Now you remember, these are some of the examples given, but you remember the difference between apogamy and apospory, which are the part of apomixis. Very simple, you have to remember. Gamy is what? Related to gametophyte. So gametophytic tissue. And spory is what? Related to sporophytic tissue. And everyone knows that gametes, they are haploid in nature. Sporophytic tissue is diploid in nature. So when gametophytic organ or the cell, that means what? Either male or female, whatever you consider, egg cell, right? Then it produces embryo-like structure, but without fertilization. That is called as apogamy. Whereas apospory means what? Sporophytic, which is the sporophytic tissue, diploid in nature already, endosperm, nucellus, that is diploid in nature, right? So when sporophytic tissue gives rise to embryo, then that is called as apospory. And when the gametophytic organ or the cell gives rise to embryo-like structure, that is called as apogamy. Very simple. You just remember with the help of names. Apogamy, from gametophytic organ or the cell, it is produced. So, and the apospory from the sporophyte. Now, what is the role? Again, we will discuss about it. The main categories of apomixis. These are recurrent apomixis and non-recurrent apomixis. Very simple and little differences there that you have to remember. Embryo sac generally rise either from an archisporial cell or from some other part of the nucellus. Then that is called as recurrent apomixis. I'll read the next also. Okay, non-recurrent. Then you will come to know the little difference, how you can find and remember the difference. The embryo arises either from the egg by parthenogenesis or from some other haploid cells of gametophyte through apogamy. Plants produced by this method are generally sterile and do not reproduce sexually. Remember this point. So, this is what the difference. Embryos, in case of apomixis, that is recurrent, it is developed from the archisporial cell and from some other part of the nucellus. And you know, nucellus, all cells are diploid in nature. So that's why this is recurrent apomixis. Whereas non-recurrent apomixis here, either from egg that is developed, embryo is developed by parthenogenesis, or from some other haploid cell. So egg is also haploid. So this is what haploid, and that's why the plants produced by this method are generally sterile and do not reproduce sexually. So this is the recurrent and non-recurrent apomixis. Now the next is what? Advent to embryonic. Remember, these are the few terminologies which you need to remember only about in this chapter. Embryos may develop from somatic nucellus or integuments along with normal zygotic embryo. Now see, little different difference is there again here than the previous apomixis. Here, embryos are developed, but what? Along with, I have
Hello, good morning everyone. येत नाही ऐकू ते recording चालू. Madam is going to join, then we will start with waiting of 5 minutes. Am I audible? Okay. My voice is clear. We will wait few minutes so that Kotwal madam is going to join to admit your friends, those who are coming late. We will wait 2-3 minutes and then I will start. I think all of you revised whatever we have covered last time. Last time we covered all the equations of the velocity on a banked road. For plain horizontal road, the maximum surface speed, that condition is there. For banked road, Both the conditions are there. Minimum zero speed is obtained as well as maximum speed that you can drive any vehicle. And again, when that angle of banking is exactly 90, in that we are considering the minimum speed velocity. Minimum velocity is required to maintain that path. Okay. Hello. What happens? I am going to close this meeting because I got message from madam. All of you, please log out and again join. I will start that new meeting immediately. No one is going. Please log out yourself. And after 2 minutes, as soon as I start with the new meeting, please join. Please log out because let me, I can see still 7, 8 names. Aishwarya, Janvi, Ketan, Kumar. Please log out so that I will start with the new meeting and again log in quickly within 2-3 minutes at 11:45 or up to 11:50. I will wait. I will wait till 11:50. All of you, please log out so that I will start with new meeting. Vineet, Shreyas, wait for 5 minutes so that I will stop this meeting. And you have to join with the same link only. till 11:50 and then I will start. Hello, good morning. We will wait till 11:50, 2-3 more minutes, then I will start so that your friends are able to join.